All right. Morning, everybody. So for the first time in 43 or so episodes, I forgot to hit the record button on Chestnut Chat. So our live version from last Friday is not available, unfortunately. So we've gotten our group back together. We are going to cover the material as closely to what we did last Friday as possible. What you missed, Russell showed up again. He has kept his perfect attendance alive through these Chestnut Chats. Our farthest attendee this time was from Thailand, but that's Brian Roth, who travels a ton. So I guess that still counts. We had Thailand, Finland, and several people from Canada. What you're going to miss out on is the frantic Teams messages that kept popping up on my screen as we were trying to get the YouTube live stream to work. So you don't get any of that, which may or may not be a good thing. But other than that, I think you're going to get a pretty standard presentation, a pretty true recreation of what we did on Friday. I am joined live here on a Tuesday morning with Jared Westbrook, our Director of Science, Vasily Lakoba, our Director of Research, and Jean Romero-Severson, who is recently retired from Notre Dame. Jean, is that a fair statement?
Last couple months, yeah.
Last couple months, all right. And then eventually we hope our other guests from Friday will record and we can paste those in. So we were also joined by Jim McKenna, also recently retired from Purdue, now a private consultant. And then we also had Tom Clack from the University of New England and Han Tan from the University of Maine. Debbie Delmer is TACF's chair of the Science and Tech Committee. She will be joining us for the beginning of this program, as will Will Pitt, the president and CEO of the American Chestnut Foundation. So this is a session where we had a lot of guests, a lot of moving parts. So relatively difficult to recreate. Again, even more motivation for me to make sure that record button is hit early in the future. With that, I think we'll get started. And just to get things off and running, this particular program is going to be about TACF's recent decision and announcement about how we have made the decision to stop developing and no longer supporting the deregulation of the Darling lines. And it's primarily because of the several red flags that we're going to cover in pretty good detail during this particular presentation. One of the main reasons we've decided to do this is because we've had a lot of evidence that have allowed us to make a science-based decision. And so this goes back to the values of the American Chestnut Foundation: optimism, patience, science-based decisions, innovation, integrity, and collaboration. These were adopted by the TACF board back in 2017. And I think all of the aspects that roll into this decision are really related to these values. Working with a tree species, especially something in tree improvement, you have to have patience. You have to have optimism. Many times the results are not something that's going to be achieved in your career, and something that you have to be able to have really long view of in what you're doing. With that, we've gained a lot of skills and experience through this work with the Darling lines and many of our collaborators, again, many of whom are here on this program. Some of the experience gained is just the knowledge about how crucial field tests are to understanding how good a product is for trees. A lot of the early results that we received showing how Darling 58 at the time, what we thought at the time was Darling 58, was effective was primarily due to tests on leaves or in the greenhouse. And then some of the more recent results that we've received are from field tests. And that's really where the true test is of whether or not the material is going to be sound in the field. And that makes sense. So, we want to make sure that for any other products that we have coming down the pipeline, we do those field tests and install them as soon as we can and across a wide geography with large sample sizes and get 12 to 24 month results at a minimum before moving forward. we think we have seen a definitely an increased acceptance of biotech for forest health, and that's pretty exciting. So while Darling is not something that we want to continue to develop, we do see biotech as a really important piece of the larger work that needs to be done for forest health restoration, not only for American chestnut, but for other species. And again, this is a decision that also is tied to that. We want to make sure that whatever product is deployed is going to be something that keeps people's trust in biotech and people's uh, acceptance for that technology. We want to make sure it works and is viable and is safe in the environment. Um, both us as an organization, our partners, and the regulators have a greater understanding of that process, of the regulatory process, and how it can apply to trees being released for forest health and widespread distribution. This was the first time that that process had been tested with a forest tree meant for release, for wide scale release. So there's a much better understanding by the regulatory agencies and by us, the American Chestnut Foundation, and other related organizations as to what that's going to look like both now and in the future. We've standardized our processes for various other testing work to minimize confounding variables. Things like, so Tom Clack, who's here with us today, he's done just incredible work doing highlight speed breeding and pollen collection, and we've been able to replicate that in other locations as well. Our small stem assays and other inoculation techniques in the field, how we collect the observation and phenotypic data, a lot of those have been standardized in a much better way to allow us to get better data and better analysis on the back end. People are more excited than ever to go out and collect wild type American chestnuts. We've expanded that library, which is great. Again, mentioning the highlight breeding and just ongoing expansion of our partnerships, which is so crucial to a program as big as this. Restoration of forest tree species has huge temporal, spatial, technical breadth. You need more than just one organization, more than one PI, more than the career of a single PI in order to really have success in this kind of program. And so the more that we can work with multiple partners across, again, a great geography and with expertise in multiple different fields, the more likely we are to have success in the long term.
Well, hi, everybody. I'm Debbie Delmer. I'm chair of the Science and Technology Committee. And my job at the beginning of all of this is to start by telling you just very briefly, what are the concerns about the Darling 58 tree that made the Chestnut Foundation decide to stop working on it and stop its support for the regulatory process. And also, why did it take so long to make that decision? So let me just outline very briefly the process of discovery and say, first of all, there was no one single thing that happened that said, wow, this is it. We learned a lot along the way, very slowly, just a little bit here and there. And sometimes what happened in one place didn't seem to happen in another location. So there were a number of reasons why this took so long. One of the things to point out right away is that the OXO gene that encodes the oxalate oxidase protein is the expression of that gene is driven by a promoter or a DNA region that tells it to be turned on all the time at very high levels. So there's a lot of OXO in these trees turned on all the time. And sometimes it's been known that that can create yield penalties or other phenomena that go on that are not so nice for trees. So it's possible that some of the things we're seeing has to do with the fact that that promoter is driving expression all the time. Some very early hints that we could, things weren't quite normal were with seedlings of D58 and Vasily saw in the greenhouse. There were some what called mimic lesions. They look like little infection spots, but it's actually not really an infection, just lesions that made the little seedlings sick. They did eventually often recover from it, and it wasn't seen in every greenhouse in every place where these seedlings were grown, though it was seen also at Penn State. So it was more than just at Meadowview where this was seen. That was worrisome. And then as the trees got older and were grown up in the field, there were other worrisome signs. First of all, there was variability in small stem assays that we're seeing more than one would like in terms of resistance. The trees often, at least I want to be careful not to say how often, we don't have good statistics, but in some locations it was pretty clear that there was reduced growth of the trees. And if you look at that middle picture of the trees with the red lines, the red line indicates on the left the height of American trees without the OXO gene, and the red line on the right shows you the height of the those that contain the OXO gene. So you can see a clear growth penalty that can range anywhere from 15 to 25%. And that can be serious if it accumulates over time. So that was one thing. Another thing, at Purdue, where the trees were grown over the course of several years, these trees, and there's a picture on the left of an OXO plus tree, a transgenic with OXO, have quite a severe canker. And so there was real variability in trees that can carry the OXO gene. There was a very great variability from no resistance to very strong resistance. And that's worrisome because these trees should not show such variability. There also was variability in the level of the messenger RNA that was produced by the gene, and that leads to the expression of the genes. So those things all were worrisome. Even maybe more worrisome for considering Darling 58 as a restoration tree is if you look at the lower right hand part of this slide, you'll see a little map of a genetic cross. Those of you who took genetics in high school or college would recognize something like this, where you cross an R, big R with a little r, a tree with a big R allele of a gene, and with a tree that has a little r. In this case, this would be an OXO positive with a big R and an OXO negative with a little r. And what you normally would expect to see is that 1/4 of the gene of the progeny from that cross would contain 2 OXO genes. Half would contain only 1 OXO gene, and 1/4 would contain no OXO gene. But in fact, this was Tom Fleck's work, and he's also been funded by the Chestnut Foundation. And what he has found is that only of the 1/4 that are expected to contain both double R, both copies, one from the mom and one from the dad of the OXO gene, that there almost none were found. Out of 40 expected, only 1 was found. So most of them, appears to be lethal to have 2 copies of the OXO gene in Darling 58. So that is very worrisome, and they'll talk about that later, the guys that worked on that. Finally, there was, and this is also a probably potentially serious in the regulatory area, there was a mix-up very early on at SUNY in terms of what event has been being characterized. And when you try to make transgenic trees, you end up with little seedlings. Each one has an independent insertion of the initial transformations, and each of those is called an event. So one event that was selected as the first and best was D58. It had high expression, and the sequence of that and its location on chromosome 7 was shown and sent to the regulators. But very soon after that, apparently, there was a mix-up in pollen, and then the pollen of another event called D54 was then sent to all of the collaborators and was used also by SUNY, and all of the succeeding results was for a different event, and even the results that were sent to regulatory were for D54, it's called. And D54 does have a problem that it's inserted into a functional gene right in the middle of a gene that is involved in salinity tolerance, or it looks like it could be involved in that. So those are all worrisome things. That was only recently found out by work of that will be talked about later in this talk. So I don't want to take too much time. I just want to say that you can see that this happened over time. It's a number of different things, no one of which might have been enough to be worrisome, but taken together really looks like a serious blow to future research on Darling 58, 54. And the reputation of TACF is at stake here. And so too is the promise of genetic engineering, because when it's done right, it really is a powerful tool, but only when it's done right. And we need to follow the science. And although it's disappointing in this case, we have to be honest and careful in how we present research. And SUNY has to make its own decision what it wants to do with Darling 58. And apparently they haven't made that decision yet. Anyway, we always knew this would be hard, but we have a lot of other interesting things going on, and we'll be talking about that over the course of the spring. So I'll leave it at that. Thank you.
So frequently asked questions. How did this happen? How did we figure out that Darling 58 is actually Darling 54, and what happened to make that switch? So in chatting with our colleagues, it appears as though way back when that initial T0, so the clone, the Darling 58 clone, was producing pollen in the growth chamber, someone went in to grab pollen from that and grabbed pollen from a D54 instead. That pollen was put on a couple of American chestnuts, created the T1 generation, 2 trees, and those were the parents and then great-grandparents and grandparents of everything that's in this testing program. So that's how it happened. It happened right at the very beginning of the program. The next question people invariably ask, well, if all you have is D54, why don't you just go back to D58? A couple issues with that. So the first is that there's only one, maybe, D58. There might be a handful. And as far as we know, they're only in the plantlet stage. They might be in a tree stage. They're back at the T0 or T1 stage. So you'd be starting over with any of this diversification and review of Darling 58. And the reason that we don't think that that's worthwhile, that that's not worth our time and resources, is because those Darling 58s, like Darling 54, are on a constitutive promoter, that 35S promoter, again, which we'll go into greater detail about. But we think a lot of these issues with the canker blowouts, with the high variability in resistance, with the growth lag, we think a lot of that is due to this constitutive promoter in particular. The third question that we generally get is, well, so TACF is abandoning transgenics. And that's not a question, that's a statement. Is TACF abandoning transgenics? And no, that's not the case. TACF still supports biotech methods, including transgenics, for restoration of American chestnut. All we're saying is constitutive OXO expression, particularly with the 35S promoter, is not likely the way that restoration populations are going to do best and perform the best and in a competitive manner in the woods and in the forests of the eastern US. We want to look at inducible OXO expression, and especially in concert with some of the breeding material we've worked with. We want to look at cisgenics, taking genes from related Castanea species and getting those into American chestnut, possible gene editing technologies. And of course, we continue the traditional breeding and fungal control work that has been ongoing while we've also been testing Darling with our partners. So again, these are red flags. I just wanted to give you an analogy. Steve Hoy works with me here at Penn State, and he's our North Central Regional Science Coordinator, and he spent a good deal of his career as a hotshot out west. And I really like the analogy of a red flag. And in wildfire, a red flag is a warning of the highest alert. It says conditions for extreme fire behavior are imminent. And so again, these issues that we're going to talk with you about are red flags. And it's not just one, it's several of these red flags that have popped up. And it lets us say, well, do the benefits of this product outweigh the risks? What are the risks to current and future restoration and commercial chestnut populations? And then are those benefits there? And obviously the benefit would be high growth, it would be blight resistance for sure. And with those benefits no longer really on the table because of what we've seen as part of the red flag, we're not so sure that some of these penalties that we're seeing with this construct are really worth the again, we don't see a lot of benefit of deploying this population because the original reason for trying to get these trees out, the blight resistance and restoration competitive capacity, getting these trees out there for ecological purposes doesn't appear to be there. And again, just to reiterate, this is one pathway that we investigated, and there are many other pathways out there that we'll continue to investigate. I want you guys to stay tuned for next month, January 19th. Our Chestnut Chat is going to be about these other pathways, where we are in these different pipelines. We'll have additional guests here on Chestnut Chat in January to discuss that with you. So I hope you'll tune in next month as well. I promise to hit the record button. And hope you guys will join us for that. With that preamble, I'm going to turn the program over to Jared, who's going to go into a little bit more detail. Jared and others who are going to go into detail about these red flags and what it means.
Hey, everyone. I'm Jared, the Director of Science with the American Chestnut Foundation. And just kind of wanted to step back and just talk about the oxalate oxidase gene. This gene has been used in a variety of contexts, like in transgenic crops, including chestnut, to basically make these plants more resistant to fungi that secrete oxalic acid. And the oxalic acid, in the context of chestnut, it is secreted by the fungus into the bark, and it suppresses the defenses of the tree. It signals the tree to have cell death, and it creates these canker, so the oxalic acid is a key pathogenicity factor for the blight fungus. And so our collaborators at ESF, Bill Powell, namely, had this insight, which I think is a good insight, to use this gene to improve the resistance of American chestnut, insert that gene into American chestnut. And so over time, what we've seen is initially the cankers on these darling trees tend to be superficial and blocky. They have this very distinct look to them. You see this kind of blocky canker, and a lot of times they're swollen, but sometimes we see this canker, some of the cankers become not superficial, and the resistance does not seem to be holding up over a long time. This one thing to note is this gene is found in a lot of plant species, things that we eat every day: bananas, rice, barley, sorghum. So this isn't going to be a risk in terms of like human health to have this gene in chestnuts. So, it's safe, and it's probably not going to increase the plant pest risk, but the way that the gene is expressed may not be most effective for restoration. Next slide. So a little background again. The gene is expressed by way of genes are kind of expressed in trees by a sequence in front of the gene called a promoter. And some promoters are called constitutive promoters, which means that the gene is expressed all the time. In the case of the oxalate oxidase gene in American chestnut, there's a promoter from a cauliflower mosaic virus called 35S, which is a constitutive promoter that's commonly used in a lot of other biotech crops. It's usually used at the beginning just to see if you turn this gene up, does it have an effect on whatever trait we're interested in? In this case, we're interested in blight resistance. So it's usually used early on in the research process. There's other promoters. There's a lot of, every gene has its own promoter, and some promoters only respond and turn up a gene's expression in response to a stimulus like blight inoculation. And so there are other lines out there, for example, that only turn the OXO gene on when the tree is wounded. And typically that's how trees get blight, is in through a wound. But there are many other promoters out there that potentially could have a very, and turn up that gene and confine that expression of that gene to tissues that are infected with blight or wounded. So that would reduce the cost, I guess, of expressing the gene, is by confining it to those particular tissues. Next slide. So one of our main hypotheses, a working hypothesis, is that this high expression of the oxalate oxidase gene in all tissues at all times throughout a tree's life is creating a cost. It's like a tree is having a fever. It's an overactive immune system that's occurring all the time throughout its life. It's leading to these growth reductions, and maybe leading to the gene being turned off in the tree over time. So this is our working hypothesis, and this is, again, it's a hypothesis. We'd have to see through different lines where we have constitutive expression versus inducible expression, whether this is actually occurring, but this is what how we're interpreting the data that we're seeing. Next slide. So initially we had done I had first started working with Darling 58, or what now we know as Darling 54. In 2018, we had some permits, and we did bred this pollen containing the OXO gene onto some of our backcross trees at Meadowview Research Farms. And we inoculated them with blight, and we looked at how much, what percentage of the trees survived 12 weeks after inoculation with blight. And we compared it to our backcross trees, American chestnuts that are susceptible, Chinese by American F1 progeny, and some Chinese chestnuts. And we see that the trees containing the oxalate oxidase gene survive at a rate of about 55%. The backcross trees are in this distribution, and we have a lot of backcross trees that are susceptible. But at the tail end of the distribution, we see the most resistant families have similar resistance to the oxalate oxidase, the trees that inherit oxalate oxidase. And we find that these OXO-positive controls are less resistant than Chinese chestnut. So the OXO gene definitely improves resistance, but it doesn't seem to improve resistance as much as Chinese chestnut, at least in these initial seedling assays. Next slide.
So this is the data from Indiana. This is our first year of crossing in 2019. We produced just about 240 seed from 10 families. It ended up to be about 46% positive for OXO. We pre-tested the seeds. And then what you're seeing here on the left, that's the canker area. So the length by the width of each canker, and you can see a highly significant effect. The positives have a numeric mean that is a lower canker, and some of them were quite small. We also included the canker rating, the 1 through 5 rating, where 5 is highly susceptible, 1 is highly resistant, and you can see the distribution there. So canker area sometimes doesn't correlate with how that phenotype looks. And again, highly significant difference, but you see the widespread in the variation. Next slide. Yeah, and so these kind of show truly the extremes. The tree on the left, this is in late November when we rated everything. We inoculated the first week of July. That's a beautiful reaction. And we didn't have any cases of any misses either. So everything we inoculated, we got a canker or a highly resistant response, like the tree on the left. The tree on the right, a little more typical for a pretty bad canker. That would be a canker rating 4, and regardless of the area. And then the one in the middle is actually one of these cankers too. We have seen a few like this, just bizarre, not a normal high highly terrible canker. That is a 5, and where we would probably say 5+. Okay, the next slide. Yeah, and this spread too. Jared took these photos when he visited in October. Yeah, these are from October now, which is good too. We find that we do need to, Sarah and I have discussed that through the years. We need to run these ratings for more than 12 months. It's so much better to evaluate the following year rather than just November, 6 months, 5 months after inoculating. So here you again, you see the wide spread. These are all OXO positives. The best tree, top left, the A tree, the B tree, very good too. That would be a canker rating of a 1. So we've now thinned the block down. We've only left about 16 trees, and we're hoping that they remain as highly resistant as A and B. So we shall see. Yeah, we're going to continue to follow these now. They're all set and thinned out in the field. All right, next slide, and I think I'll turn it over to Jared.
Another red flag is these are data from ESF. We are starting to see what we saw in these data were increasing variability in the expression of the oxalate oxidase gene over multiple generations. And so these bars represent how much the gene is being turned on in the stems of American chestnut. And the founder generation is the D58 tree. The first generation are the first generation of crossing to wild American chestnuts. And the second generation, you start to see variation in that expression of that gene from higher than the founder tree all the way down to 0. And this variation might be explaining why we're seeing the variation in expression of the gene, might be explaining why we're seeing the variability in the cankers. This is kind of a big red flag, because what this implies is that the gene is being turned off by the tree. And as it's turned off, it can get, this is progressive, the way the tree turns off the gene. And so it's possible that the tree, over time, the trees could lose their resistance. And I'll turn it over to Gene to explain a little bit more about gene silencing that's typical with 35S promoter.
All right. Well, for those of you who don't know what a promoter is, Jared touched on this. A promoter is the switch that turns on the gene. A constitutive promoter is like the light switch that most of us have in our houses. We flip on the light, we go about our business. If we never flip that light off, it just burns until the light bulb burns out. An inducible promoter is more like a motion sensing light that only comes on if someone's in the room. So in this case, what would be good is a wound inducible promoter. Something that turns on only if there's a little wound, which allows the blight to get in and start its pathogenic process. The cauliflower mosaic virus promoter is a promoter from a virus that expresses any gene it's in front of all the time. There aren't very many constitutively expressed genes in a normally functioning organism. And what happens in biotech frequently when you use the cauliflower mosaic virus gene and you track its expression over time is that the plant that it's in, that plant sensing stress will silence that gene. And the longer that plant lives, the more intensively it will silence that gene until eventually it can succeed in silencing every single copy of that gene it has. And that silencing can persist over generations. So this is a flaw in using constitutively expressed genes. As Jarrett said, they tend to be only used in the very early stages where you're testing the efficacy of the gene that you're interested in. Because trees live longer than the year that corn and soybeans live, a constitutively expressed promoter is not particularly desirable on the landscape. A wound-inducible promoter would be better, but again, Mother Nature is full of subtleties, and wound-inducible promoters, some are better than others. Some come on very early and then fade over time. Some will come on much later and stay on at much higher levels. So this is the second layer of research in using biotechnology to achieve a certain result, deciding on which of many promoters to use. And that is the stage we're at now.
At this point, it's important that we recognize some of these other red flags that we're seeing in the phenotypes of the Darling 54 trees. So one of these red flags are the growth penalties that we've been observing in a variety of settings that seem to come along with the constitutive expression of the OXO gene. So on the left there, you have a picture in an orchard of 2 rows of trees. You can see there's a red line over each row. And in those rows, all of those trees are siblings, so they have the same parents. Now, because there's only one of those parents, the one providing the pollen, that can contribute the that construct with the OXO gene and the 35S promoter, only roughly half or a little less than half of those trees end up inheriting that gene. Otherwise, they get their contributions from the 2 parents. And you're seeing on the right those trees that did inherit the gene. So those are the OXO positive siblings, and then on the left you're seeing the ones that did not. And evidently, as you can see there, the ones on the left that did not inherit that gene construct are growing taller. And this is something that has been noticed now in a variety of settings, whether it's in an orchard with trees that are a few years old, whether it's just in the first year or 2 of growth in nursery stages. You can see on the right there, there's 2 plots summarizing heights of trees in an orchard at Purdue. This is the second transgenic cross at Purdue. And you can see the first plot there that says 2021. So in 2021, again, these are comparisons of siblings. The negative trees are growing taller than the positive trees. So there appears to be roughly a 15% growth penalty that's statistically significant of the inheritance of that gene. And then in those same trees, you look over to the right in 2022, by then that gap in growth has expanded to 25%. So when you notice something like, first of all, a 15% reduction across many trees, and then in the next year it expands to 25%, that's certainly a pattern that you want to track over time, and we're intent on doing that because it's very important to see that there's not going to be this constantly expanding gap between the trees that do and those that do not inherit the transgene, which is supposed to confer blight resistance, but as we've seen so far, even that is very much in question. And the reason this is important, of course, is that these trees might be growing in an orchard or in a nursery, but in terms of restoration populations, those trees are going to be growing in a forest setting, and they're going to have to compete with other trees, other vegetation for light, for other different types of resources. And it's very important that their growth is not reduced by expenditures on the expenditure of energy on the transgene's activity. Now, of course, you do have trade-offs in biological function of organisms, but things like a 15% reduction growing to a 25% reduction in just a year in an early life stage, that is a big red flag, and that's something that we have to keep investigating further. Next slide. Okay. So going further with another red flag, building off of the reduced growth. So again, this reduced growth has been seen in a variety of settings. Here we're highlighting records of this that we have in nursery settings across greenhouses compared with other different environmental variables. We're able to see across the board this difference between the OXO positive siblings and the OXO negative siblings. Again, I want to highlight that these trees, when they're within one experiment, we made sure to compare trees that are offspring of the same 2 parents. So the background genetically is very, very similar. The difference is the inheritance of that transgene construct. So again, seeing reduced growth, sometimes pretty heavily reduced in those that do inherit the transgene. In this case, and also injury occasionally under stressed conditions. So while these trees do not always show these symptoms, they certainly do under certain stresses, and this has been replicated in a variety of lab and nursery settings. Down below at the bottom of the slide, you can see that sort of characteristic leaf curling and browning and general disfigurement of the leaves, which eventually progresses in this type of injury under stress and leads to stunting, less photosynthesis, and sometimes death of the seedling within the first year or two. And yeah, this is of course something that we're still investigating as to what is the exact biochemical mechanism for why that happens, but we've seen this pattern repeatedly enough that this is a red flag and a concern. And again, so these are elements of poor performance of the trees inheriting this construct for non-blight reasons. So Jared covered some of the problems with reduced and progressively reducing blight resistance. These are problems that are actually not even caused by the blight fungus. On the left, we just have a plot of from another experiment, again, siblings, so same parents, half of them inherit the transgene, half do not, of severely reduced survival just within the first year in the nursery of the OXO-positive D54 siblings. So again, all of that amounts in concert to another red flag and something that really needs to be investigated further. First few years for a tree like chestnut is really nothing. It's infancy. And so if these problems are occurring this early, it's important that we track them now and make sure that we work on solving them and before we proceed with any kind of restoration or reintroduction. All right, so again, all of this goes back to the fact that our ultimate goal is not just the production of trees that show some type of potential blight resistance in a lab setting, in a nursery setting, in even in an early orchard setting. The goal is to be able to reintroduce American chestnuts to the forests of Eastern North America, have them survive, have them go from seeds to seed-bearing trees, have them reproduce, open pollinate, and have those populations independently grow and sustain themselves over time, eventually amounting to what we would call restoration. So in order to do that, it's vital that these trees have competitive ability in the forest setting. Not only are they going to have to compete with other trees, with their fellow chestnuts, with other species, with an increasing number of competing vegetation in the form of invasive plants, and a variety of other stresses that come with changing forests through the past century and through the century to come. So knowing that and seeing these red flags this early on, it's very important that we step back from launching into large-scale accelerated production of this particular transgenic event because we are seeing these red flags. And it's important that we go back to some of the more experimental phases in order to develop lines that will be worthy of reintroduction and have a chance of success in actual restoration settings. Next slide.
So this is reporting on a collaborative effort between the University of New England, where I am at, I'm Tom Clack, and my graduate student, Virginia May, along with Han Tan, a geneticist at the University of Maine. And we've been pursuing homozygosity in darling plants, meaning that the plants would have two copies of the transgene, the OXO gene. We want that because then the pollen could pass on a copy of the gene to every wild type flower that we pollinate. So we've been crossing darling moms and darling dads in our speed breeding lab at the University of New England. And as you see in the table, the expectation is that 25% of the offspring of the crosses between two darlings should inherit 2 copies of the gene, and 50% would inherit 1 copy, and 25% would inherit no copies. So about a year and a half ago, we began to push our plants to produce viable burrs. And early on, we were sending immature burrs, about halfway developed or 2/3 of the way developed, to ESF for embryo rescue. And out of those first 18 that we sent to ESF, 2 came back from PCR testing as homozygous for the OXO gene, lower than expected, but indicating it's possible, but unusual. And then over the last year or so, we've been putting our darling pollen on darling female flowers in the lab and taking them all to full nut maturity in about 13 weeks, and then growing them out into seedlings, and then sending that plant tissue to Dr. Tan at the University of Maine for testing for homozygosity. And we found that so far out of 39 that have been tested, only 1 is homozygous. So mostly the result is lethality. And we can conclude that it's possible to produce homozygous darling on darling crosses, but it's rare, and we'll continue to breed and test.
So that's much less than the expectation of 25%. Suggests that possibly that the homozygous, when the gene is inherited in homozygous state, there's some kind of deleterious effect to where that tree can't develop from an embryo into a seedling. And the other thing that they found during this process of testing for the homozygosity was that they were expecting the gene to be located on chromosome 7, that we know that in Darling 58, the location of the gene is on chromosome 7 in between at the very end of the chromosome, in between a few genes. So it's not disrupting any native gene, but they were doing, in the process of testing for homozygosity, they were not finding the gene where it was supposed to be. So they did some additional tests where they actually were able to get whole genome sequencing data, and they mapped the gene insert to chromosome 4, which is going back to the petition for the deregulation of Darling 58. They found that the producers were also using another line called Darling 54, which had the 35S OXO gene inserted into a native chestnut gene. And that insertion actually created a deletion, so that gene is no longer functional. And so when we're seeing this homozygous lethality or reduced viability of these homozygous offspring, we still have this question of whether the disruption of this gene is causing that problem, or if it's a dosage effect of having 2 copies of the OXO gene turned up at a high level. So that's an ambiguity that we have, but certainly the homozygous lethality is an issue going forward. Next slide. So, it's a nice thing. We can still generate trees that are what are called hemizygous, which means they inherit 1 copy of the OXO gene from one of their parents, but we haven't been able to recover these homozygous trees, which inherit 2 copies. It'd be really nice to have orchards in the future that would be all homozygous, because all their kids would also be homozygous. And so that would mean that we wouldn't have to test any of the seeds anymore. We could actually just harvest open pollinated nuts from those orchards, and we would know that all of the nuts inherited the gene, and we would no longer have to go through this laborious process of coring into the nuts and putting in, testing to see if the gene is present or not. So this would be a major benefit in the future of large-scale production of blight-resistant seed. So I think that this is although we can still get inheritance of the gene, I think the capacity to form homozygotes is an important feature that we have for a restoration population. Next slide. So why not just go back to D58? And you know, there are, again, there probably are a few D58. We don't know how many are actually out there at this point. And we know that there's at least one in sort of tissue culture, small tree phase. So we don't know if the pollen is available, and there might be a few these trees of the D58 offspring in the field. But I think that a lot of these issues are related to the 35S constitutive expression, the gene silencing, the growth lags, the lack of homozygosity. All of that is probably related to the constitutive expression. So if we are having to start over, we might as well start over with different lines that are currently under development that use different promoters that only turn the gene on in tissues that are infected with blight or wounded. And so there are, you know, a lot of different promoters that we're trying at this point, and we want to see which of those promoters is most effective at enhancing the blight resistance without creating these additional costs to the tree. Next slide. So we're working with a collaborator at the University of Georgia, Scott Merkel. He's one of the people that really worked on the methods to basically culture chestnuts from embryos and clone them, basically. And this is a way that you can insert genes into the American chestnut genome through these embryos. So Scott, you know, has these methods developed that basically take about 2 years to go from seeds that we collect in the field, immature burrs, to culturing those burrs in petri dishes and multiplying them in some, like turning them, like cloning them out and growing them up into trees, basically. And that whole process takes about 2 years. And he's been working on a number of different new OXO insertions using different promoters. And so those are all in the pipeline at this point. So it may be worthwhile just to wait a few years to get to these other additional lines that are not Darling 58 that we have that we can use for testing that have different promoters. Next slide. So what does success look like for OXO? What we're looking for ultimately is we want to be able to, you know, get to the point where we're field testing these new lines, and we want to see minimal canker expansion at least 12 months after inoculation, no evidence of gene silencing, minimal growth lag, similar survival and vigor between transgenic and non-transgenic, and the capacity to form homozygous offspring. And with constitutive OXO, at least with Darling 54, we're not really seeing any of those yeses coming through. With inducible OXO, we don't know. And that's why we need to go through this process of going through some of these field trials and testing for gene silencing and testing for homozygosity. And that's going to be an important thing to do before we go through with regulatory review of these new lines. Next slide.
And so again, just to reiterate the kind of alternative pathways we're looking at, and that are continuing from even during when we were examining the Darling lines, and what we'll cover in much more detail next month. You know, this is one of the biotech solutions that are available and that have been investigated so far. You know, some of those biotech solutions are really in their infancy. Things like gene editing or even mutagenesis. You know, actually mutagenesis was looked at in the 1950s, but has also been ongoing in other species. That might be something that we want to investigate moving forward, but which has not yet been started. But some of these other things like inducible expression or stacking with other genes or looking for genes from Castanea that might be able to be used in a cisgenics kind of a program versus transgenics. You know, these are things that are ongoing and that might be available for review in the relative short term. Things that we've also been doing consistently for several decades in the traditional breeding realm. You know, backcross breeding is something that we continue to look at. It didn't quite work the way as it had originally been hypothesized, but there are still some really nice trees out there. And through the great work that Jared's been doing with collaborators at Virginia Tech, Hudson Alpha, and other locations, we have some models which allow us to pick the best trees, breed those together, and enhance resistance in that population. We've got resistance from large surviving Americans that we're looking at. Phytophthora resistance is being increased in those populations. And then looking at ways to continue to control the fungus. Hypervirulence is still out there. Again, not quite as easy to control or release as had been hoped, but still an option. There's also other ways of looking at the fungus to try and get it controlled. So we'll have some folks who are working on these different solutions who will join us in January to talk more in depth about what's been going on, what the potential application is, and a little bit more about timeline and when folks can expect these solutions to be in research and practice in the field. Again, it really comes down to the importance of collaboration and working with all these different folks. You know, I can't thank folks like Jean and Han and Tom and Jim for helping us, you know, evaluate the current status of this material and also help us chart a course for the future that will eventually see success. And I want to thank you all for showing up and allowing us to do this work. It really isn't just the academic and science and research side of things. It's about optimization of techniques in the field, and then it's about deployment out in the field. And we've learned that you can't do that as a singular organization. You can't do that as a singular institution or as a singular PI or researcher. It really takes, these are just a few of the many collaborations we enjoy, and just a few of the thousands of volunteers who come out year after year to help do the various types of work that we continue to do. So I hope that you guys will join us in January to dive in a little bit more deeply about the work that is to come in the next few years. And I love ending on this quote. This is something that Lisa Thompson, our recently retired CEO, would always end her slides with. I wish I could come back in 100 years and see my trees. And that really is sort of the timeline that we're on. We're on tree time. And so we just have to keep remembering that this is not a person people timeline that we're on in terms of large scale restoration, and the work that we do will eventually be realized. We see small snippets of that timeline in the work that we do, but eventual success will be for generations to come. With that, we'll start some questions and see where we're at. So again, you know, because this isn't quite live, we did have the Q&A saved from Friday, so we're going to cover a few of the more popular questions that we had and some of those that we did answer live. There's a lot of questions about, do I have Darling 58? Do I have Darling 54 in my yard? Do I need to cut it down? Jared, do you want to cover that one?
No, you don't have Darling 58 in your yard. To get that tree, to have these trees, you need a permit from the U.S. Department of Agriculture. And we have a number of sites that we have specifically permitted with trusted collaborators that can watch over these trees. And one of the conditions of these permits is that they can't flower. They can't spread on their own. We can inoculate the trees in their first few years of growth to evaluate their resistance and evaluate their growth. But we still need to do this under permit from the U.S. government. So the trees that you likely have are either wild American chestnuts from our we have a seedling sale every year. We have volunteers that distribute wild type American chestnuts to volunteers and enthusiasts. We also have a member seed distribution that we distribute seed from the Meadowview Research Farms primarily, they're hybrids that have improved blight resistance, but still they have intermediate levels of resistance and still variable. So that really is, you know, that's a gift of membership. We're not trying to sell you trees and guarantee their blight resistance, but rather it's a way to keep people engaged as we continue to improve this, you know, the American chestnut resistance over time. So that just keeps all of us engaged in the process to actually have tangible trees that you can look at.
Thanks, Jared. I'm going to pitch this next one to Vasily. Number one, kind of building on that, do I need to remove the trees that I received from TACF? And should I keep nurturing them along and keep them?
Yeah, good question. So to the first question, do you need to remove the trees that you do have, which are, again, as Jared said, not D54? You maybe have backcross trees, or maybe even have some pure Americans. Those are your trees on your property. You can do whatever you like with them, but there is certainly no um, uh, nothing saying that you need to remove them for any reason. There's no need to do that unless you want that tree gone yourself. And in terms of keeping them around, and which hopefully you do choose to do that, as we've been highlighting in terms of what we've gained from all of this experience, all these years working with the American chestnut, it's very, very important to continue conserving native genetic diversity, because eventually when a line does come along that is worthy of restoration, reintroduction, you now would have potential mother trees for crossing onto. So certainly hold on to your trees if you're willing to. Please do that. There's certainly no need to at this juncture to take them out.
Thanks, Vasily. Jean, I want to give you this question. There's a two-parter. And Jared covered it a little bit, but I think it's worth repeating. There are a lot of questions about what is a promoter and what are the different promoter options besides 35S, and tell me more about a wound inducible. So what is a promoter? Maybe a little bit more about 35S and what's known or not known about it, and then wound inducibles specifically, and any other type of promoter you might want to cover.
Right. The promoter, every gene in a higher plant, higher animals too, has a switch in front of it. We call that switch the promoter. Okay. And these switches function differently for different genes. So a constitutive promoter is a switch that's very much like the light switch in your house. It's on. Once that switch is flipped, it is on all time, 24/7. In nature, there aren't very many genes that are expressed constitutively. That's what we call it. In nature, genes tend to be a little more regulated than that. So the wound-inducible promoter is a promoter that acts in a little more subtle way, like a motion-sensitive light that only comes on when there's people in the room. The cauliflower mosaic virus promoter is a constitutive promoter. Early in the process of studying a gene that you might want to use, you use that promoter because it's always on, because you want to see if the gene you're interested in does have the phenotypic effect that you want it to have. So you want that expression always on. The difficulty with the cauliflower mosaic virus promoter, and we found this out very early in the private sector when I was there, is that you pay a penalty for that. Even in annual crops like corn and soybeans and sunflowers and cotton, there tends to be a yield penalty or a growth penalty when you use a promoter that is always on at the level that the cauliflower mosaic virus promoter is on. So early on in the private sector, we learned to investigate different promoters that were more subtle in action, that were only turned on if the insect chomps or the fungus invades. And those promoters tend to have fewer, if any, performance penalties. Okay, now in our context, performance isn't yield. Performance is, can this tree live out its full life and be sustainable in the forest? So that's why we're interested in how it grows, how it survives as a seedling, and also in the efficacy of the gene. When you put cauliflower mosaic virus into a plant, a promoter like that into a plant, and this will happen even in corn and soybeans, that super blazing hot constitutive promoter, the plant senses that, hey, my energy is being drained off here. There is a mechanism of defense against that in plants and in humans as well. And that mechanism of defense is, we're going to shut it off. So the plant will, over the course of its growth, begin to shut off that cauliflower mosaic virus promoter. In a perennial like a tree, that shut-off process can continue for a long time, and it tends to persist across the generations. So once that gene is silenced, it tends to be silenced for the tree's whole life, and probably the whole life of its descendants. And this is definitely not something that we want, because then you have no efficacy at all.
Gene, one thing that we haven't talked a lot about is I know that there's interest from the commercial industry. There are some really nice European cultivars or Euro-Japanese hybrids even that don't have full blight resistance on their own. And I know that there's interest in trying to get some of these blight resistance technologies incorporated into those commercial lines. So yield is going to be of importance to them, not necessarily the American Chestnut Foundation. But I wonder if you can talk a little bit about yield with 35S. I mean, how is that balanced? And especially for chestnut, it's going to take at least 5 years to even look at what the yield's going to be, if not 10 to 15, when you have that sort of starting to reach that maximum height of yield in a chestnut tree. So any thoughts about that and things that we should be looking out for from that perspective?
Well, I think based on the the private sector has been doing this for 30 years. And a lot of the information they have gathered, they don't necessarily publish everything, but what is very well known is that the cauliflower mosaic virus promoter is infamous for a yield penalty. So now when investigating, let's say, cis genes, like I identify some genes from Chinese chestnut, for example, or Japanese chestnut that are contributing to resistance, you would never take that cis gene and plunk the cauliflower mosaic virus promoter in front of it. You wouldn't even start that because of its well-known reputation for a yield penalty. Okay. And I don't want to seem negative about what our collaborators did. It's natural it makes sense to use it at first in the early part of the process. But now I think it would be wise not to even start with the cis genes, because the possibility of a yield penalty, well, you never know until you put it in the tree and you actually look. The phenotype is the truth after all. But I would strongly advise against it. Don't even use that promoter. You use a different promoter from the start. Now, there are other constitutively expressed promoters that don't tend to have such high expression levels. There are other constitutively expressed promoters that let's say have less follow-on properties, but still any constitutive promoter in a perennial, especially where you're looking at yield. When a tree is filling all those chestnuts or filling its ear or its soybean pod, an immense amount of carbon skeleton is going into that process. And any other process that's going on that's draining off that energy, it would be like when that's draining off that energy is what is the tree going to do when its energy is being drained? It's going to damp down its yield, because preserving its life is the most important thing, because this is a perennial organism. Okay, even in corn, as the seed is filling, if the promoter is constitutive, there will be less seed fill. There will be a yield penalty. So that's why that's why it is wise not to invest in that particular promoter, and hopefully any constitutive promoter, for a product that's intended to, one, yield enough seeds to sustain itself, exist competitively on the landscape. And in the case of the chestnut growers, where they're going to take care of the tree, but where they're interested in yield, this would not be a good way to go for them. If I were giving out this advice to the chestnut growers, that's what I would advise.
Thanks, Gene. And so Jim, I'm glad you're here. We did have a question on Friday, and I'm going to repeat it here, about the strange looking cankers and that sort of blocky behavior. Do you only see that in the transgenics, or have you seen that elsewhere?
Yeah, there were some cankers that were totally unique. In fact, I told Caleb, because it was his first experience to be able to rate these. And yeah, I told him, I said, Caleb, this one here is Frankenscience. There is. It's interesting. I was hoping, I'm still hoping, that some of the trees that really resisted blight and didn't have much growth yield could still be durable. There's only one way to tell, and that is leave them and continue to evaluate them. And so that's what we plan to do, for 2 years at least, right? We want to do that. I mean, who knows what we can learn from some of these. I made notes on the data sheet. Jared, I sent you that too. Jared should have that. And I believe I've sent it to Eric Carlson too. So everybody has that. And, we're going to share all the data with everyone, whatever could be learned from these, right? And because we've got them too, we've got 2 more fields. But no, the blight, and we had natural blight coming too into the seedlings. We must have had 6, 7 or so, Jared. We saw that, and there were more with the minuses. They were small cankers, nothing too big. But no, it's just it's a footnote for a few of them, and of course I noted those just in case there's a molecular discovery that could be made with that, right? Yeah, so we have that material.
Thanks, Jim. And so, Jared, I think there's a question here for you, which is what is to become of the remaining D54/D58 trees? Jim, has said, further research is warranted, and I think, that's the case. TACF has stated we don't intend on developing them any further and don't recommend them for release and deployment. But what do you see as far as researching these trees and where we can take that?
Yeah, so I think the key distinction is that we're continuing with some research, as long as it's not going to be draining a lot of TACF's resources, but like inoculating trees that we have in the field, we're going to continue to do that and continue to monitor the trees that we have inoculated, like in Jim's plots in Purdue. So we do have a number of field trials that are in the pipeline now at numerous permitted locations. So we want to continue doing that research. And one of the things that I'm really interested in is we've crossed some darling trees with large surviving American chestnuts that have partial resistance to blight, and some of our backcross trees that have partial resistance to blight. And I'm wondering, is there an additive? Is there a stacked effect of stacking these genes? OXO with native genes for chestnut blight resistance. And we really have to see, like, over time, are we seeing the durability of the resistance improve when we stack OXO with these other mechanisms of resistance? So that's an ongoing research question, and I think we can investigate that preliminarily with darling lines, but we would want to move into inducible lines as well to see how when we have this gene expressed in a different way, does that effect of these other genes also change at all? So we have to test in these different new lines that we're developing as well.
Thanks, Jared. There's a couple questions here that I want to take on. One is about the regulatory activity. The question is that halted as of now? At this time, it is not yet halted. We know that the folks who submitted the petition, ESF, intends on continuing on with the regulatory process, and that's something we no longer support just because we don't see this as being advantageous for restoration. The other question is in terms of the patenting of future chestnut lines that we might want to work on. So, one of the great things about the work that Bill Powell did with Darling 58, and the intention was to leave this in what can be referred to as the genetic commons. I really like that term, and that's certainly the intent of the American Chestnut Foundation. TACF does not support for-profit commercialization of any of these materials, and we don't support patenting of that work. There are some ways that you can protect intellectual property that protect both the producer and the person receiving the material. So there's something called a germplasm agreement that TACF uses. Love it or hate it, it is something that does help protect both us and the producer. And this comes down to a lot of question about, well, what would happen if this material had already been deregulated, if Darling 54 or Darling 58 had already been deregulated and distributed into the wild? What would you do then? And so a couple of the controls that we have with material that's sent out. And this is a key feature of the germplasm agreement that TACF uses, says, and we use that primarily with backcross material. But what that says is, we want to ensure the viability, the appropriateness of this material for the ecology and the greater environmental purposes. So having a document like that sets the stage for the people uh, using the material. There's no guarantees assigned with this material, and that's stated pretty explicitly in that kind of document. And then also allows us to have some visibility of where material goes and eventually gets planted. We also have an online database called Dentatabase, where we track our distributions and where stuff is planted, and it's also available to private landowners and our research collaborators to be able to track their material and where that goes. So, that's not a patent. That's not a way to commercialize the intellectual property or to commercialize the tree. It's a way to be able to track it and know not only where the material goes in case there ever needs to be some sort of QA/QC callback, but also to track the amount of work we've done. How can you track and really quantify the amount of trees that have been planted towards restoration? And when you look at there's some 100 million acres of suitable habitat out there, you're going to have to be able to track what your progress is, not only toward planting, but then also the spread of that material as it goes forward. So, those are the kind of tools that we have in place moving forward, but certainly not in the for-profit realm. We are adamant as an organization that this project remain in the nonprofit realm. And kept in the genetic commons, like I said, as much as possible. All right, let's see. Do we have another question here? All right, so last question I think we'll take here for this session is any new developments with hypervirulence? And then a related question, anybody working on learning more about the blight itself, different varieties in different parts of the range? Jared, I think I'll pitch this one to you.
So the hypovirulence, there has been a development of a genetically engineered blight fungus that is able to transmit. First of all, let's talk about what hypovirulence is. It's basically where the blight gets viruses itself and it becomes less virulent as a result of having this virus. So it's a way to kind of control the fungus from spreading, and it's worked pretty well in Europe. And the way the virus is transmitted is the fungal hyphae from the chestnut blight fungus have to be able to merge and transmit the virus. And there's genetic incompatibilities that prevent the spread of the virus. So University of Maryland researchers actually developed a strain of the blight that has more compatibilities with a lot of different genetically diverse strains of the chestnut blight fungus that can transmit this virus. So it should hypothetically be able to control the virus. And what they found initially was that they had to treat every individual canker to control the virus or to control the fungus. And they had to do this multiple times. So on a restoration context over a long, huge area, this is probably not a viable method to control blight for all the remaining trees out there. But to keep trees alive in an orchard where we can actually treat them, it potentially has some merit. The thing that we've seen that's been a big roadblock is that the U.S. government, USDA, has some reservations about introducing this genetically modified fungus into the environment. So they want us to prevent it from spreading. So it's very difficult to get permits to actually use this in an operational setting at all. So we've actually kind of put that on the shelf right now, using those hypovirus strains, those super donor strains at least, for controlling the blight, the modified strains. Now, in terms of understanding the virulence in the fungus, we have a collaborator that will speak next week, Bruce Levine, who is looking at what genes might be involved in the fungal virulence and what genes the fungus might be using to turn off the tree's defenses. And if we can figure out how those genes are working and what genes in the tree they're actually interacting with, then we could put in other genes from other trees that are not susceptible to blight to actually block those genes' actions, the virulence genes' action. So that's another avenue that we're exploring, and we'll talk more about that in January.
Thanks, Jared. So lots to discuss in January. You can see why we split this up into two parts. Still a lot to cover here as we unpack and research what is going on with Darling and what we can learn in terms of silencing and, like Jared said, with stacking of genes. That will be ongoing, and we'll certainly report that as soon as we have results to share. So stay tuned for next month, where we'll talk a little bit about what's next and share some of the collaborators we've been working with and we'll work with in the future. In the meantime, Jean, Jim, thank you guys, Tom, Han, Will, Debbie. This was a fantastic session. Thank you for re-recording it with me as well. Like I said, in the future, I'll be sure to make sure that auto record, that I don't hit stop on it, but we don't have to redo this again. Vasily, Jean, Jared, you guys have anything else you want to say for the go to the audience?
Have a good holiday.
All right.
I need to go bake 12 dozen Christmas cookies for my very large extended Wisconsin family. So I'm saying goodbye
Fantastic.
now.
Thanks,
Right.
Jean. I really appreciate
Bye-bye.
it. And yeah, have a happy holidays. We'll see you in the new year.
Happy and safe, you all. Yeah.
Happy holidays.
Right.
Thanks. Bye.
Bye.